In this part, I have to say the penicillin classes overview. Penicillin classes कोणकोणते आहेत? Narrow spectrum, wide spectrum. Narrow मध्ये आहे natural penicillin आणि very narrow spectrum penicillin, anti-staphylococcal penicillin. Then वाईड स्पेक्ट्रममध्ये दोन आहेत, एक तर एक्सटेंडेड स्पेक्ट्रम, स्मार्ट वाईड स्पेक्ट्रम आणि एक व्हेरी स्मार्ट वाईड स्पेक्ट्रम पेन्सिल. जे वाईड स्पेक्ट्रमचा जो पहिला क्लास आहे तो that includes gram positive as well as gram negative bacteria, and the last class it includes anti-pseudomonal penicillins. Okay, now types of resistance against penicillin, natural and acquired ones of the bacteria. Resistance in bacteria for penicillin, then acquired against penicillin in detail we have to see, and then we have to talk about the pharmacokinetics, what the drug is doing, what the body will do to the drug when it will be taken inside. That is the pharmacokinetics of penicillin. Here we can see the overview, narrow spectrum and the wide spectrum. Narrow spectrum natural ones are penicillin G and penicillin B. Next is the anti-staphylococcal. Those are nafcillin, methicillin, cloxacillin, dicloxacillin and flucloxacillin. Then wide spectrum are extended and anti-pseudomonal penicillin. In the extended we see only two, amoxicillin and ampicillin. Amoxicillin, it is very much absorbed in the GIT, and ampicillin, it is poorly absorbed in GIT. Also there are anti-beta lactamase drugs. What is this? To add it along due to susceptibility. I don't understand that. Okay, I will see. These are the anti-pseudomonal penicillins. Those are the piperacillin, the ticarcillin, then carbenicillin, azlocillin and mislocillin. Anti-beta lactamase drugs to the added. ये काय लिहिलं रे यार? अच्छा. The anti-beta lactamase, the beta lactamase inhibitors that are added with these penicillins. Okay, and so those are clavulanic acid, sulfabactam, and tazobactam. Okay, I got it now. To be added along due to susceptibility. To be added, okay, yeah. They were. In the overview again we are seeing the resistance against penicillin, natural and acquired. Natural is cells that are without peptidoglycans. All of them they have resistance against penicillin. What is basically penicillin? It will go to the PBP and it will prevent the transpeptidation. So anything that does not have a peptidoglycan, it will definitely have. Anything that does not have a peptidoglycan, it won't be killed by penicillin. And those are human cells, mycoplasmic bacteria, mycobacteria, chlamydia and pseudomonas. Okay, so they do not have peptidoglycan and hence they are okay. Next is acquired resistance. So they have acquired the resistance by producing beta lactamase. It will destroy the penicillin and the penicillin will not work on that. Decreased permeability. Okay, in the gram-negative bacteria, they don't have the permeability for some penicillins, and they have very narrow porins that only allow some penicillins to act in it. The permeability. Increased efflux pump. What it will do the efflux of the penicillins that are coming, and it will not let the penicillin work. And next is the alteration of the protein binding protein. This protein binding protein, it will get altered from its genetic material, and then its structure will be changed, and whatever the penicillin is coming, it can't bind to that altered PBP. Okay. Next, what here is shown is it's a bacteria with DNA, and it is having a gene. Okay, and that gene is producing beta lactamase. Whereas this other bacteria, it is having its circular DNA along with plasmid DNA, and this plasmid DNA is producing beta lactamase. And whenever there is signal, what we call that, a transduction, conjugation, whenever there is conjugation between two cells, that is bacterial sex we call that, and what will happen, this one bacteria, it will form a sex pilus, and its plasmid will be transferred from one bacteria to another, and that another bacteria will start producing beta lactamase, because the plasmid that was transferred, it had the genetic material for the production of beta lactamase. So plasmid mediated and the chromosome mediated is there, and this thing, when it will multiply, it will give its progeny the gene for beta lactamase. And when one bacteria has to give its plasmid to another bacteria, completely another bacteria. And that is also possible, where what is possible, the beta lactamase giving each other is possible. Okay, now here we can see the efflux thing, and here is the altered PBP that alters the transpeptidases. That means, yeah, same. Then talking about, here, we can see, so there are two types of resistance. Why is resistance happening? Because there's continuous modification of the drugs. In the bacteria, they develop resistance against beta-lactam drugs. What are all the beta-lactam drugs? They are penicillins, cephalosporins, then aztreonam and carbapenem. Natural resistance. Naturally resistant to the action of anti-beta lactamases due to absence of the peptidoglycan in the cell wall. So काही bacteria आहेत त्यांच्या cell wall मध्ये peptidoglycan नाही आणि so they are naturally resistant to the action of beta-lactamase. Okay. Here we have to understand that Pseudomonas, it's a gram-negative bacteria. It does have a peptidoglycan, but small porins are present, very narrow porins are present that make them resistant to the beta-lactam drugs. Mycoplasma does not have a cell wall. Mycobacterium, they have thick layer of mycolic acid. And chlamydia, they are the bacteria that multiplies inside it. Chlamydia are the intracellular bacteria, and fungi, the cell wall is present but not made of peptide, it's made of chitin. And human cells, they do not have a peptidoglycan, they don't have a cell wall, so yeah. And if we were having, if and whichever cells that produce the beta lactamase, they have a peptidoglycan. Action of anti-lactamase. What these penicillins, these beta-lactam drugs, what they do is they prevent the transpeptidation. They prevent the peptidoglycan layer formation in the bacteria. And if a certain bacteria has to produce resistance against that particular penicillin, that means that penicillin, it should not destroy the cell, the organism. And if that organism does not have a peptidoglycan, then that particular penicillin, it can't do transpeptidation there. It can't do that particular penicillin, it can't prevent the transpeptidation there. Okay, and hence those bacteria, those organisms, they would be naturally resistant to the penicillin. And these organisms we have seen. Mycoplasma, it does not have a cell wall, so the penicillin, it won't come, it won't prevent the cell wall formation there. Okay. Now there is acquired resistance. And wait, what this particular sentence is saying, that the naturally these particular things are naturally resistant to the action of anti-lactamases due to the absence of the peptidoglycan in the cell wall, whatever. Now the acquired resistance, some bacteria produce enzyme that break the beta-lactam ring, beta-lactamase. And yeah, if beta-lactamase producing gene is part of bacterial chromosome, these bacteria will produce beta-lactamase. Bacteria and all its progeny derived cell will resist the beta-lactam drugs by disrupting them. This is the most common mechanism of the acquired resistance, by producing beta-lactamase. And plasmid-mediated beta-lactamase production when extra-chromosomal. Okay, when bacteria with chromosome and plasmid multiply, they can transfer them to many other bacteria. This form of bacterium has a great mechanism of protecting against the beta-lactam antibiotic, which has many locked-in plasmids in its genetic makeup, which protect it against beta-lactamase drugs. Now the bacterial conjugation. Both bacteria have bacterial chromosomes, but one bacteria has a plasmid. This plasmid has one gene that codes for beta-lactamase and another that expresses special proteins in these bacteria. These proteins come to the surface of the bacteria and a small tube-like structure that starts growing and will attach the membrane of the other bacteria, which will receptively provide a hole. When sex pilus is produced, plasmids open and pass through and reach the other bacteria. Former bacteria were resistant. Since the second bacteria received plasmid, it also became resistant to the action of penicillin. This process is called as resistance through bacterial conjugation. This leads to resistance in other bacteria, making the situation worrisome. Conjugation is far more common and far more efficient to transfer resistance from one group of bacteria to another. Conjugation. Alteration in genetic structure reduces the permeability to penicillin and clavulanic acid. Some bacteria alter the genetic material and produce porins, tightening the porins and resulting in penicillin resistance. Efflux pumps. Some bacteria develop efflux pumps and actively take the anti-beta-lactamase drugs. And what are the anti-beta-lactamase drugs? Those are the anti-staphylococcal drugs out of the cell wall, thus inhibiting its action and becoming resistant. Efflux pump. And these particular penicillins, the very narrow spectrum penicillins, they will be kicked out. And these are chiefly present in the gram-negative bacteria. Okay, wait. Anti-beta-lactamase drugs. Okay, the ones that are the clavulanic acid, that was the beta-lactamase inhibitor, no? Then the sulbactam and tazobactam. So what they will do, they will kick out, they will bind to that particular beta-lactamase, and it won't let that destroy the beta-lactam ring of the penicillin, of the beta-lactam containing antibiotics. Now, okay, wait, I now can relate one thing here. The natural resistance to the action of anti-beta-lactamase. Anti-beta-lactamase, the beta-lactamase inhibitor. Next thing is regarding the alteration in penicillin binding protein. Some bacteria alter penicillin PBP, which disturbs splitting or attachment of beta-lactam ring, hence making it resistant. Example is methicillin resistant Staphylococcus aureus, MRSA. What they do, they alter their PBP and hence the methicillin, it can't come and destroy them. Another example is Streptococcus pneumonia and Enterococci. Vancomycin works on MRSA, always remember. Because what was vancomycin doing? It was doing the glycosylation, right? It was preventing the glycosylation. No? So, still the cell wall synthesis can be inhibited in MRSA due to vancomycin. Here is some Staphylococcus bacteria altered the enzymes involved in transpeptidation glycosylation. So this is one more thing that the bacteria have done, like they have completely altered the enzyme only. Then the pharmacokinetics of penicillin. Some can be given orally, some biparenterally, and some can be given both orally and parenterally. So the ones that only can be given orally are penicillin B, amoxicillin with clavulanic acid, and indenyl carbenicillin. Parenterally, penicillin G, ticarcillin, carbenicillin, piperacillin, and ampicillin and sulbactam. It is together called as unacin. Unacil. And the rest of the penicillin can be given both orally and parenterally. So ticarcillin, carbamazepine, piperacillin, penicillin G, and the last one is ampicillin with sulbactam. Okay. So penicillin in depot form, that is the long acting, it is given with procaine penicillin, and the benzoate penicillin. Penicillin G is complex with these, okay, and it's given IM and it works for 3 to 4 weeks. It is given IM to patients with rheumatic fever. Okay. We had seen that particular amoxicillin was there now. It was very much good, it had very much good absorption into GIT, but ampicillin it didn't. So amoxicillin completely absorbed, good for injection when amoxicillin required systemically, but not good option for GI infections because the GI infections will still thrive. Ampicillin is poorly absorbed through GIT and prefers to stay longer, hence efficient for GI bacteria like Salmonella, Shigella. Antistaphylococcal penicillin absorption is affected by the presence of food. And what are the antistaphylococcal penicillins? Those are the methicillin, napsylin, cloxacillin, dicloxacillin, and flucloxacillin. And hence it should be taken either on an empty stomach or 2 hours after meals. Penicillin is not lipid soluble and does not pass through the cell membrane. Okay, always remember, penicillin has got what? Okay, it is not lipid soluble and does not pass through cell membrane. So not all intracellular bacteria are attacked by penicillin. Penicillin is well distributed in ECF and leaks into blood, interstitial fluid, and many other body fluids. Penicillin cannot pass the blood-brain barrier unless the barrier is broken. In meningitis, in encephalitis, and inflammation, penicillin does not enter bones normally but can do so in osteomyelitis and other infections. Penicillin can cross the placenta but is not toxic to the developing embryo. Most penicillin is not metabolized much and is excreted from body via kidneys, passing through urine except napsylin, which goes to the liver. Organic acid secretory pump in PCT, proximal convoluted tubule, there is an organic acid secretory pump. What it does, it transfers organic acid to the lumen of the nephron, and 90% of the penicillin use these pumps, which go to the nephron and urine. This is important in penicillin with shorter half-life. Why? Because drugs like penicillin G are given with another drug, and this other drug, it occupies these pumps. So our penicillin, it does not go into the nephron, rather it is it again comes to the peritubular capillaries, and thus the action of penicillin G is prolonged. One such example is combination of penicillin G with probenecid. It is not fully absorbed because probenecid, it binds to the organic acid transport mechanism, sending the penicillin back to the body. So probenecid, it binds to the organic acid transport mechanism, and it won't let the penicillin get into the nephron. Next is the penicillin can appear in milk of lactating mother and can cause diarrhea in infant. It is also secreted in saliva. And to revise resistance against penicillin, natural acquired resistance mechanism are by producing beta-lactamase. It will disrupt the beta-lactam ring. Plasmid-induced beta-lactamase production. Plasmid would make the beta-lactamase, then to bacterial conjugation, the plasmid will be transferred, and the other bacteria would also get what? This particular beta-lactamase, and alteration in the genetic structure to penicillin and clavulanic acid. Okay, I don't remember this. Alteration in genetic structure to penicillin and clavulanic acid. Okay. In genetic structure means its porins will be narrowed and tightened, and these penicillin clavulanic acid, they won't permeate, and hence if they can't come, then they can't destroy us. Then next is efflux pumps that will efflux, that will keep out the they will keep out the beta lactamase inhibitors. So wait, the efflux that actively take anti-beta lactamase drugs out of the cell wall. Yes, they keep the anti-beta lactamase drugs out of the cell wall, thus inhibiting its action, becoming resistant, and chiefly in the gram-negative bacteria. So wide spectrum. When we talk about the efflux pumps, we talk about the clavulanic acid and all those beta-lactamase inhibitors in the wide spectrum. Okay, and alteration in the PBP. Yeah, if PBP changes its structure, then penicillin can't come and bind to it, and transpeptidation won't occur in all of these cases. Pharmacokinetics: penicillin can be given orally, intravenously, or both. It depends on which one it is. Some penicillin can be given in depot, and they are not lipid soluble. They cannot pass. So penicillins, they are not lipid soluble. Penicillins cannot pass blood-brain or blood-brain barrier if intact, and bone barrier if intact. Okay, most penicillin excreted out of body through urine. However, shorter half-life ones need additional drugs that bind to the organic acid secretory pumps and prolong the stay of shorter acting drug in the blood. They can cross placenta. Okay, yeah, fine.